Here we are, we're doing Genetics 2. I'll slide that over there to the corner, and I've subtitled this lesson Gene Actions, and then I said interactions because you can interpret it both ways, how genes interact on the same locus. There's always two copies, two alleles. Sometimes they're the same, sometimes they're different. And then later on we'll talk about how genes at one locus might influence genes at a different locus. But I do want to give out a shout out to Gregor Mendel, who worked with peas, and many people credit him with being the father of genetics. And I know some of the interactions that I'm going to be describing can be contributed to him. I think like the first one I'm going to talk about is dominance recessive, and I think he looked at that. He was interested in how progeny get their genetics from their parents. So shout out to Gregor Mendel, father of genetics. Okay, so let's look at our first gene action, sometimes called interactions. Dominance / recessive. Sometimes you'll see it dominant recessive. I've seen it called complete dominance and complete recessive. Okay? Trying to give you a bunch of the jargon so when you see something it doesn't floor you. Okay, so in my example, the capital A is going to be the dominant allele, and the lowercase a is the recessive. So the three genotypes, now remember those are genotypes right there, and we're keeping it very simple. Those three genotypes will all have the same phenotype, because as long as you have one copy of large A, then it's going to show that phenotype. Now remember, this is all at the same locus. We've got to remember the dominant recessive. So A is dominant over little a at the same locus. Remember, there's two alleles at the locus, at any locus. So then here I'm going to bring in the little a. Only in this case do you get something different phenotypically, right? So this is where the recessive gene is going to be expressed only then in its homozygous recessive state. Then we're going to find out what little a codes for. Here we're only going to find out what large A codes for. But you should know that although these are all phenotypically the same, the homozygous dominant can only pass large A to its offspring, whereas these two genotypes will give some of their offspring the little a, right? So you got to remember that. So sometimes little a might be a disease, and so these individuals here would be carriers, but the disease would be not expressed, and the disease would only be expressed with the homozygous recessive state. Well, we're on to the next type of gene action, and I labeled this incomplete dominance. You might also see it as partial dominance. So let's say, and I'm going to use an example in chickens. A makes black chickens, and then a little a makes white chickens. Okay, I'm going to put that down there. So large A, black, and maybe what I'll do is I'll write that in here. Okay, so if I didn't say so, I'm talking about plumage in chickens. So large A gives you a black plumage. Little a, white plumage. Then, if we do the heterozygous state, and I know I'm being a little redundant because large A, little a is exactly the same as little a, large a. But if you're not familiar with this stuff, you might say, where's that other one? So anyway, that animal has a plumage of like a bluish gray color. And gray, I saw it spelled Grey too, so maybe I should have done it that way, but that's not important right now. So that's incomplete dominance. The you get some blending, I guess is the best way to say this, a blending of the two alleles to give you a third phenotype. Okay, so like here's one phenotype, here's the genotype, and here's the phenotype. Here's another phenotype, and here's another phenotype, the color. So that's incomplete dominance, sometimes called partial dominance. Okay, now we're talking about codominance. And there are two main examples that I could use. One that I'm just going to mention and not show you, and that's the ABO blood typing in humans. That's an example of codominance. And I should probably say codominance is where you have two alleles at one locus and both alleles are expressed equally. So there's no dominant recessive thing. They're both, you could say they're both expressed. And the example I'm going to use, since I like cattle, is the roan coat color in cattle. Roan is a blending of red and white, and it ends up being roan. A roan cow, you can look at the hairs, and some are white and some are red. So it makes a very neat example. So R codes for red hair. And so if you have a large R, homozygous R, large R, you get red hairs. If you have large W, you get white hairs. Now in this example, the two alleles, there's no like little r or little w. It's just large, capital R and capital W are the two alleles that can fit at that locus. So then, if you have one large R and one large W, in either way, right? I mean, I'm doing here, I'll shake these. Those are both equal. And if I say both alleles are expressed, well then lo and behold, those animals are going to have red hairs and white hairs. And that's going to give you, get rid of that, I don't need that over there. That's going to give you the roan. So that's an example of codominance. Both alleles are expressed, and that's different than incomplete dominance that we had just discussed. So codominance is a very interesting gene action. Okay, ready to talk about overdominance. Years ago, I guess they called it superdominance, but I don't believe that's terms used anymore. So overdominance. Well, I'm using my capital A, little a example. So we're still talking about what's happening at one locus. In fact, all the discussion in Genetics 2 has so far been just talking about the one locus alleles interacting at the one locus. The reason I'm making a point there is there's going to be some time when we talk about how one locus might affect another locus. Okay, so you know that this is homozygous, capital A, and let's say that genotype has this type of phenotype, right? So genotype, large A, you get a production of, let's say, a B1 antibody. I'm giving you a theoretical example here. Okay, well then, if you look at the homozygous little a, notice I'm not saying dominant recessive, I'm just saying little a, those animals with that genotype produce a B2 antibody. Who knows what we're talking about there? It's a theoretical example. Then in the middle, which I'm jiggling up and down, we have large A, little a, and little a, large a. I'm just doing that for completeness. That's the heterozygote state. Well, those animals with that genotype produce B1, B2, and B3 antibodies. So the production of the heterozygote state exceeds either of the homozygous states. So another way of saying that is overdominant. Sometimes they'll say the heterozygotes are more extreme than the homozygous state. Another example I saw was like, let's say we're looking at the fitness of an animal, and higher the number means it's better fit for its environment. Let's say little a has a fitness score of 1, and then large a has a fitness score of 2. Well, the heterozygote state, you might think would be someplace between it. No, this would be like a 3 or 4. So when there's heterozygosity, that means they are exceeding either of the homozygotic states. That's a lot of tongue twisting there. That's overdominance. Okay, here we are. We're talking about pleiotropy now, where one gene affects multiple traits. That's what I'm trying to depict here in my figure that I created. I didn't have to draw anything, so it looks pretty good. So here it is. One gene at one locus might be coding for an enzyme. And this is actually a real example in humans. I didn't want to complicate it by giving details, but it's a great general example. One gene makes an enzyme. So if there's a problem with the gene, then you affect multiple traits. And that's really pleiotropy. One gene, sometimes they'll say one allele, probably either one is pleiotropy, where you have the one locus, one site, one genetic site there, one gene affecting multiple traits. So like in my human example, there's an enzyme that's made, I'm not going to name it, but it might affect, let's say, skin pigment. That would be my trait 1 example. Mental agility, mental ability, maybe I should say, for trait 2. And trait 3, body odor. So like 3 quite different traits, all relating back to one gene product. That is pleiotropy. And I'll have to say, I've got a pretty good example there. Okay, now I'm talking about polygenic traits. Well, whenever you see the prefix poly, that means many, and polygenic, the genic there refers to the genes, so many genes, and I guess it's going to be at least 2 or more. I've got an example down here of 3 genes working to make one observable trait. Sorry for that tongue tied there. For example, one phenotype. So what's an example? Well, it could be the height of a dog, height of a horse, the weight, mature weight, could be blood pressure of the horse or a dog, whether an animal has diabetes, and also human skin color is a polygenic trait. Okay, we're going to talk about epistasis. It's a type of gene action or interaction. Not sure what's the best term. Usually they say gene action. And epistasis, is where an allele at one locus. Now, pay attention here because we're talking about different loci now, which we haven't hardly before. We probably did when we did the polygenic, yes, but most of the dominant, recessive, codominant, incomplete, all that kind of stuff was at the same locus. So here it is. An allele at one locus masks the effects of an allele at a different locus. Okay, so that's epistasis. So be careful there because now we're talking about different locations. And the best example I have is what's known as the white gene in horses. And I think one of our lessons in genetics is going to be at least a little introduction to horse coat color. There are some main genes that give you the main colors, but then there's what's called style genes, where you get the blazes and the socks and all kinds of stuff, and it takes quite a bit of studying. But let me tell you about the white gene in horses, how it is epistatic. So here is a genotype, right? Large W, little w, and then I've got these blanks. And you know, and later on we'll talk about what letters go in there, but what I'm saying right now is if a horse has a large W and then a little w, it doesn't matter what the rest of the loci have, right? because this one large W, capital W, is epistatic over all the other genes that code for coat color. I'll give you a little hint here. Like this next group we'll be talking about when we do horse coat color is the gray gene. Well, even if the horse was going to be gray, with that large W being epistatic, the horse will never be gray. So now, if the horse is little w, then if it was a gray gene here, and we'll do it a capital G, then the horse would turn gray, and the little w would not interfere. So it's only when there's one large W. So you might say, what happens if there's a homozygous large W? And I'll put it over here. Oops, sorry, can't drag it with my laser pointer. I can drag it with this. I'm going to put it over here. If a horse has that genotype, if it has two large W's at that locus, the horse dies in utero. It's usually never born. I'm not sure if it's 99.9%, but anyway, that's called a lethal white gene. If it's in its homozygous large W state, that offspring with that genotype is never born. Okay, so it's kind of interesting. So let me get back here. I want to talk about how if a gene like this large W overrides all the other genes, it's called epistatic. This large W is epistatic. And then the ones that are overridden, perhaps a better way of saying that, are called hypostatic. So this place here, whatever the gene is, I haven't told you, but it's hypostatic to the large W. So that's epistasis. A great example, the white gene in horse is epistatic to all the other genes, the solid color genes as well as the style. It's going to be a white horse if it's got large W, little w. Okay, I'm going to end with this this term. I've got so many, I've got to make another little lesson with other things like inbreeding and outbreeding and qualitative traits and quantitative traits. But penetrance. Penetrance, and I'm not going to give you maybe all the definitions, but maybe one good one to remember is the frequency with which a dominant gene manifests itself. So what I'm saying here is that maybe just because there's a gene present, that it always doesn't manifest itself, isn't always expressed. So that's kind of like a little chink in the armor, isn't it? All this interaction we talked about. Maybe sometimes a gene isn't going to express itself. I'm sure somebody with a big genetics background could do it better, but that's my definition. Here's an example. Polydactyly in humans. Polydactyly. This is actually having more digits, more fingers. I guess it probably also counts with toes. You might say, what? Well, there are some humans that have 6 fingers. Yes, I remember seeing somebody that had 6 fingers years ago. Polydactyly. It ends up being, I think it's a dominant trait, but obviously the allele isn't present very often. But what happens is, it's not always expressed. Even with the person that has the gene, and I believe it's a dominant one, I don't know what letter they code it with, that's not important right now. But let's say that 9 out of 10 times when it's present, it shows itself. So it has 90% penetrance. Okay, and that's just an example. I'm not sure if that refers to polydactyly. I'm giving you an example, percent. Okay, so polydactyly in humans, not always present. If it's present, if it shows up phenotypically when it's out of 10, 9 times out of 10 times it's there genotypically, then it's 90% penetrance. And then, this is a term that, man, how do you say that? Expressivity, I guess. And this is if it does show itself. So now we're saying if it is showing itself, it doesn't always show itself to the full extent. So what am I saying? I'm saying maybe when it's expressed, you get a full length of finger, and it's like a normal sixth finger. Or it can be a little stub or someplace in between those two. And that's what this term is referring to. Okay, that's it. See you guys later. Bye.